Hello there, this is Long COVID Foundation Podcast. UK's first long COVID podcast channel for sufferers, researchers, practitioners and everyone who wants to learn more on COVID. Please join our group on Facebook and share your stories. And now is Dr. Valentina Vituto, Long COVID Foundation Trustee.
Hello there, it's Long COVID Foundation Podcast. Today we're gonna talk about Long COVID second wave. Why we have it, issues with it, high numbers, and the reasons behind them. This channel is for those who suffer from long COVID, for researchers and a whole host of other people who'd like to know more. And I'm very glad to say that today we're gonna have a very special guest, Professor Baranov, who is from George Mason University. So welcome, Antje. I'm really glad to see you on the channel, and I'm sure all our guests are gonna have a great time listening to you. So welcome.
Thank you so much, Valentina. I'm very happy to see everybody. My name is Antje Baranov. I'm from George Mason University. I'm professor in the School of Systems Biology. So I work with pathophysiology of various human diseases. I am virologist. So by my first trade, when I graduated from Moscow State University, I graduated with a degree in virology. So for me, indeed, came very interesting time. And I put together, because of that, or maybe because I wanted to help my friends in Russia, I put together a couple of videos, which were in Russian first, about the biology of coronavirus and the biology of the resulting disease, the methods of treating it. And to my big surprise, those videos became very popular. And this is basically why I'm doing this video right now in English, because I believe that maybe something which we learned since that could be useful not only for professionals who are working in this area, but also for everybody who got coronavirus or got coronavirus and cannot really completely recover, or somebody who has relatives with this virus and want to help them, at least by understanding of the disease.
Absolutely, Antje. And I would like to mention to our group members that Antje is running regular podcasts with her listeners, and she's getting a whole bunch of different questions, very tricky questions to answer, and she does research in the area. One of the latest papers Antje presented is talking about the reactivation of long COVID, mentioning the very, very serious case that happened in America. So I'm sure Antje would tell us a little bit more on this.
There was a recent, very clearly documented case which was described in New England Journal of Medicine. New England Journal of Medicine, one of the leading journals in the world medically. This is on par with Lancet. I think that in UK everybody knows Lancet, right? Yes, so New England Journal of Medicine is American analog of Lancet. It's also very esteemed journal. So there was a paper published in this journal, and I will describe this case to you in details. But please bear in mind that if one case is described in details, probably there are hundreds of cases of this kind which just got undetected because this patient didn't happen to live across the Boston hospital, or because this patient didn't get tested and sample didn't get sequenced during the first wave or something like this. So the case which I would describe to you in details is a case of 45 years old man. And you see, he's not very old. Yes, 45 years old. It's not an old person.
It's not in a group of vulnerable patients.
Well, he was, because he had some underlying condition, which is so-called antiphospholipid syndrome. Antiphospholipid syndrome is not that rare. It is rarely diagnosed, but it's not that rare. Antiphospholipid syndrome is one of the many autoimmune disorders which plague humanity. He came to the hospital with a flare of this antiphospholipid syndrome. But again, I want to say that antibodies to phospholipids are common in population. It's just this guy had a lot of those antibodies. That's why he sparked attention from physicians.
Mhm.
On the other hand, there are people with lower levels of those antibodies, and they just co-found on some diagnostic sheets when tests are done and largely ignored. People are not treated for that usually. So he came with coronavirus. This coronavirus was confirmed by test, and in this particular case, they took a sample and sequenced it and put it into the bank.
Mhm.
The patient was treated in hospital for like 3-5 days, something like this. Get better, get back home. Then he was home for 2 or 3 months, but during this 2 or 3 months home period, he was tested negative, so there was no coronavirus on his nasal sample,
Mhm.
but he still was sick. He was suffering from a host of problems, including cellulitis. And cellulitis, I know that many think cellulitis is some kind of cosmetic defect, like girls come to the cosmetologist, say, here is some cellulitis. I have no. In this particular case, cellulitis is really severe disease, which is an inflammation of the adipose tissue under the skin. Yes, which is severe condition. It can lead to fever, it can lead to sepsis, etc. So he was treated for the cellulitis. So even when he was home for 2-3 months, he was getting to the hospital, and that led to confirmation of his negativity for the virus. Then he got sick again, and that was again compatible with coronavirus. They took sample of the virus from nasopharyngeal cavity again, sequenced it, again treated him in the hospital for like a week or so, then again released him home. And then he stayed home for another 2 or 3 months. And in home, he was suffering again from like long COVID. Then third time he came to the hospital. That was day 148 or something of his disease from the very beginning, day 148. And then he was in the hospital till he died on the day 157. So altogether, that was 157 8-day disease, almost 6 months. So this patient died, and he died in the hospital. And when he was in the hospital, he multiple times went through the collection of the virus. He was positive again. And that includes his bronchoalveolar lavage being done. That is the procedure from which the virus is collected, not from nasal cavity, but from the lung. Dramatic procedure, but that could be done. And in his case, it was done. And when he died, his organs were sampled for virus also as well. So what we learned from this case? This was true case of long carrying the virus with 2 reactivation of the virus, 2 episodes of reactivation. So first episode was put down by physicians, and second episode proven fatal. So second time it didn't came down. When they analyzed the viral sequences first time, second time, third time, they were able to make a tree of all virus species. And that is actually the brilliant point in this paper, very, very convincing. They, on this tree, showed that the virus number 3 is direct derivative of the virus number 1 with intermediate of virus number 2. That went through evolution, and this evolution was happening within the host. And every time when evolution produced the virus which was able to escape immune system, it led to flare. And then physicians put it down, virus continued to evolve, and that came back again. So all this time, all 6 months, virus was slowly but surely evolving in order to propagate more and led to flares.
Oh.
And I think we have cases like this. It's not the only case. I should say this patient with antiphospholipid syndrome, for sure his immune system was in disarray. We couldn't say that his immune system was normal. For now, we should think that situations like this are only possible with people who are suffering for some kind of the defect in immune system. But if you think about it, there are many people who suffer from certain defect in immune system. First of all, patients with HIV, infected with HIV.
Mm-hmm.
Their immune system far from being in good shape. And yes, of course, when they are on antiretroviral therapy, then their system function more or less okay. They're able to fight viruses, they're able to get vaccinated, etc. But if this disease come to at some point out of control and to fruition as actual AIDS disease, maybe because patient was not getting medication, maybe because gaps in medication, which is very important for country where I am in United States, because not every patient is under insurance and not everybody getting antiretroviral therapy. Then that leads to the decrease in the immune function, and then this kind of reactivation may happen. Another important thing that we have natural immunosuppression states. One of them is pregnancy. So during pregnancy, women bodies suppress immune system, own immune system, in order to not to reject the embryo, right? And that makes a window for vulnerability, for reactivation. The last situation, of course, is the old age, because in old age, you know, the immune system does not function ideally. And of course, it's different for different people. So we couldn't really measure everybody with the same measure. No one ruler for everybody, because some old people have very good immune system, and some people lose the strength of their immune system already in their 40s and 50s. Very often it goes hand in hand with metabolic syndrome, obesity. But we can say that each of us with aging undergo some kind of the decrease in immune system function. And if the virus is able to be invisible in our body for a very long time, then with age we are getting more and more vulnerable to reactivation. I should say that this is not proven. It's just some derivation which I am making from this published paper and some other papers. I will name for you yet another paper which was published recently. That was a real good paper in Cell, and that is saying that in people who get mild disease, really mild disease, they didn't even went to hospital, mild or moderate disease, they do have immunity to the virus. And if you take samples of the cells, B cells, from the patient who get completely recovered, not with long COVID, just completely recovered, months after infection, 2 months, 3 months, 4 months, 6 months, those antibodies which are produced by those B cells would not be exactly the same. They would be a little bit different from what was there in the beginning. And the late antibodies in 6 months are somewhat better in neutralizing the virus than original antibodies which were there when we started the process. From the first glance, that's a good idea. That's a good thing which we really observed, because that means in 6 months your immunity only get better, right? They are neutralizing virus better. But when I read it as a biologist and virologist, that was real scary to me, because in nature, everything is happening under the selective pressure. So the evolution from not so good to better to absolutely best only happening under pressure. This means that there was something which was pressing on immune system to create better and better immune response. And that is only possible in the presence of virus. And there are two explanations for that. Or maybe, you know, we can have both explanations at the same time. One explanation is that virus will continue to sit inside. So even in the people who get the virus very mildly, very mild disease, still there is virus hiding somewhere, and our immune system get itself shaped up by the presence of the virus internally and constantly fighting the virus. This is, by the way, exactly what is happening in the bodies of the bats. So bats fight coronavirus exactly in this way. They got coronavirus, okay, then they start to live with it. Yeah, for a very long time they live with it without any symptoms. Second possibility is maybe we are getting infection all the time coming from outside. But that also not a good possibility. That means that virus is everywhere and we are getting constantly exposed and shaping up our immune system to this external influence. That's not good either. And I think that the first version is more likely, and we need to study it more. It's not a sentence for humanity, yes. We probably would be able to deal with it, but in order to deal with it, we need to really understand what's going on.
Absolutely. Well, Antje, thank you very much. You've made really great points, and I'm sure we're gonna get some more questions coming in terms of what should we do? Do we have a virus inside us and it's gonna appear in a couple of years again? So I hope this won't happen. And as you mentioned, vaccines going to protect us at least for some time. As we heard, the immune system gonna last at least 2 years. Let's hope it's gonna be the case with vaccines. But we're going to have another podcast on vaccines where we could explore different types of vaccines in different countries in more details. I would like to ask some basic actually questions to help our listeners to understand what is the second wave. Why do we have it and how different it is from the first wave?
First of all, I think that the whole thing which comes in waves, like first wave, second wave, it's a little bit of media creation rather than scientific creation. There is no scientific theory which says that viral diseases should come in waves and there should be this many waves and no more waves, or second wave stronger than first one. It's just a popular understanding and media creation, nothing more. Why coronavirus get a little bit less active in summer? This is because the virus is causing acute respiratory condition. And all viruses causing acute respiratory condition are less stable in summer situation when we have summer in comparison to winter. However, when we have seasonality, the seasonality is not extended to the same degree depending on the climate. So some countries and some localities within countries are more likely just simply because of the climate. So for example, you know, I'm Russian, as you can derive from my thick Russian accent, right? So I spent a lot of time in my childhood in Moscow and its vicinity. Moscow have continental climate. Continental climate, that means it's very severe winter, and this is dry, maybe short, but dry, sunny summer. This condition is not good for virus in summer to propagate. And the change between summer and winter is very drastic. So when people adapt to this change is causing the changes in their bodies, and this change make people vulnerable to the virus. So it's very common in the continental climate to have very strong difference between amounts of acute respiratory diseases in summer and in winter. And when the virus comes, it comes in droves. So a lot of people get sick at the same time. When we have the climate which is a little bit more mild for various reasons, for example, this is a country which is on the seashore, right? Or like in case of England, this is Ireland.
Yeah.
And this is island which is located somewhat to the north, and why it is getting not so bad climate, that's because of the Gulf Stream. Diluted seasonality. So you could have good summer, you could have bad summer. Very often you have wet summer, where there is a rain, fog. So when there is rain and fog in this situation, virus cannot die down. So of course, in Moscow, in summer, we have more dramatic decrease in virus activity in comparison to England. And that is exactly what we saw in situation of coronavirus infection. So the waves were more pronounced in Moscow in comparison to England. Now look at United States. United States is a very big country. Actually, I'm talking to you from United States, Virginia, Fairfax. It's very close to Washington, D.C. And we have long seashores, very long seashore lines. And we have southern parts and northern parts. In the southern parts, we almost have no winter. It's a tropical climate, subtropical, tropical climate. In the northern part, which is adjacent to Canada, we have strong seasonality, and this is continental climate, pretty much like in Moscow. North Dakota is not really very different from Moscow. So in those places, we have also very dramatic seasonality, but in summer, for example, Florida had peak of epidemics during summer. It really started real bad there in the July and August this year. Texas a little bit more continental, but those parts of Texas which were near the Gulf, They got it in summer as well, and then with the change in the climate, it spread to the north. So waves are not really there because they are part of the biology of the virus, but because we have seasonality, and seasonality in some cases working for us, in some cases working against us. And of course, I know that this question would come to everybody's mind, what would happen in the winter? Well, you remember any kind of pandemics, epidemics, just acute respiratory infection wave which happened during this time. And you remember that the worst month is actually February.
Absolutely, yes.
February, when it's very wet, it's still cold, it's get a little bit warmer, but you're already very tired of winter. That's where majority of people get influenza. So this would be the same in the situation too. Our hope that vaccine actually will be there and it will start to give immunity to at least some percentage of people before February come.
So what we see at present in the UK is that number are actually increasing now. And as you mentioned in the summer, yeah, we had really nice drop in these numbers. And starting from October, this number started to grow. People struggling to understand why they're getting the virus for the second time. We do have tests if you suffer from high temperature, cough. People affected back in March are getting tests now and they're again testing positive for a second time. Can this happen? Or testing system doesn't work? Why people are getting second COVID at the second wave?
Well, as you can yourself conclude from what you just say to me, yes, it can happen. And you cannot really blame it on tests. Yes, tests could be sometimes false positive or false negative, but it doesn't cancel the fact that majorities of the tests are working fine in every particular case. Otherwise, they would not be approved for the use in general population. That's exactly what regulatory organs are watching for. They are watching for tests delivering test results being accurate, meaning sensitive and specific. Yes, they do have certain percentage of the errors, but this is acceptable percentage. And when you see cases like this, which are en masse, so when somebody is telling you, oh, I have my aunt who tested positive second time, I had my brother who tested positive second time, that is already undeniably the second positive test. Now, it's not exactly the same as proven case of reinfection or reactivation, because scientific proofs require to show undeniably with scientific data that the new virus is different from the first virus which infected patient back in March. The only way to show it scientifically is that when you were sick first time in March, somebody in the hospital, doctors or scientists, collected your sample of the virus and not just quantitatively measured the virus, but showed that this virus have specific sequences. So the virus was sequenced. Its sequence was put into certain database. And then when you got sick again in October, again, virus samples were taken, again sequenced, virus was sequenced, again put in the database. And then the third scientist would come and compare first and second sequence and show that it is impossible to derive second sequence from the first one. So it should be independent case of infection. Now, if you remember yourself and your friends and relatives in the first wave, how many of you actually went through the process of collecting the virus and sequencing it? Very few.
Absolutely. And I think in the UK we also had a quite significant challenge to get a test. Most of us now try to prove that we had coronavirus back in March and we're not lying, and we had exactly the same symptoms that are under NHS guidelines now. But we can't prove because we didn't have a test. We see this happening again for the second time, and we have to prove to our health practitioners that, well, it's the second time I'm getting it. Please do something. Please check on me and do some kind of tests. As you said, nobody did it in March. Nobody can prove We had it in March and it's the second time. So people are getting into a problem. I don't know if there is any kind of solution to get proper treatment. Is the virus mutating? Is it somehow different? How people cope usually with catching virus for the second time?
First of all, I want to say that yes, we do live in the world which is far from being ideal, but we knew it before. Before coronavirus, we already knew that world is not exactly ideal. So nothing new we discovered in the process of fighting coronavirus as well. I mentioned one thing why we could not really prove the reinfection, but there is also another thing which is related to the biology of the virus. When somebody gets infected, first coronavirus start to multiply in the nasopharyngeal cavity. So it's somewhere in the mouth, somewhere in the nose. And those are the places which are easy to reach. So the nurse can go with Q-tip, collect the sample, and then profile the sample. Mm-hmm. If person is not getting to the doctor during those first 2, 3, 4 days for getting virus, and that happens when you are in line waiting the appointment with doctor, and meanwhile your disease develops. It develops to the point that it goes to your lungs or infects other parts of the body, but those parts are not reachable anymore. So say you were in line waiting for the doctor appointment for 5 days, you were sick all the time, but when you came to doctor, the sample was taken and you are coronavirus negative.
Absolutely.
Yes, the sample came negative, but if it would be taken 3 days in advance, that would be positive and you would know, but now you would never be able to know. Well, how to solve this problem? In United States where I am, and I'm a little bit more knowledgeable about situation domestically, I should say that the situation is changing because we have the types of tests which are approved for use without doctor prescription. Well, they are still going under doctor prescription, but this is going throughout the kiosks. So somebody can just drive through the kiosks. There is a nurse, and nurse just ask you 2 or 3 basic questions like, do you have fever? Do you have running nose? Like this, how long you sick? And then she's getting you the automatic prescription for the test, and then you're getting test right there. And then you receive the result of the test by email, and then this test getting into the central database and also sent to the doctor, your doctor, which you indicated in the papers. It didn't work smoothly. It didn't work exactly the same way in different states. So some states get into this horse earlier than others, but we do have the system right now in place. So situation a little bit better than it was in May and June. That's clear. Also, there is another kit which was recently approved. I should say that this kit called Lucira kit is not widespread yet. We just got approval 4 or 5 days ago, and they, from the very beginning, told us that for now the kit would be available only in 2 hospitals nationwide, one in Florida and one in New York, I believe. So this kit is use at home kit. It is still by prescription of the doctor, so they don't want people to test themselves and then just sit on the result, not sharing them with nobody. But it can go through telemedicine. So you call your doctor would not see you, but just say, oh, your symptoms are compatible with coronavirus, and then you are getting this test mailed in to your house. Test is based on so-called LAMP PCR technique. So you collect the sample yourself from your nasal cavity, you put it in the little test tube, then you do some magic, and then you put it in the device which comes together with the kit. And device is also like very small cooker, like very small cooking pot. Use the electric connection or the battery connection. And this cooking pot is one-time use. So you put this sample, you put it into this device, and device tells you whether you have coronavirus or not. And then you take the test from the device, make picture, and you send it to your doctor. So it will go without contact with doctor to the central database countrywide. And also your doctor, based on this test, will send you prescriptions.
In the first wave, we've been told that most vulnerable people may get it. So people over 65 may get it. Now with the second wave, we see that the age group become younger and younger children now getting it. Why does this happen? What has changed in terms of mutation of this virus that children have started to get it?
Well, that's very, very important and interesting question. And I should say it's not because of the virus. That's because of the people and the diversities of our genotypes and diversity of our immune response. Don't get me wrong, virus does mutate. And we know that D614G mutation did indeed spread throughout the Europe and United States, and it's replaced original Chinese strain. And this strain is more capable of infecting people. So one person with the disease infects more people on average than it was during the spread of the first original Chinese strain. But it doesn't really contribute to making the disease more severe. So they are getting basically the same disease. The presence of D614 mutation does not really preclude response by antibodies to this virus. So if you have antibodies from the previous infection, during the second infection, you still will be able to neutralize virus. But people are different. We have a lot of people. That is a news for everybody. People also have various state of immune system. Even among youngest people who should be having the most active immune system, able to fight virus real fast, even among those people, there are people who do not know about it, but they have some problems, inherent genetic problems with their immune system. And those people get severe disease. When they get severe disease, it brings attention, and then it gets onto TV. So 23 years old girl get coronavirus, died. Yeah, that's a news. When that is happening with 87 years old grandma, that's not a news, that's statistics. We have cases of people who are vulnerable to diseases, but not knowing about that, getting the disease, make the news because of that. We are thinking that those cases are more widespread. Now, what happened with children en masse cannot be explained by that, even if some children have weaknesses in the immune system, of course. But what happened is that children react to coronavirus without really producing strong immune response. And that is great. They're able to clear the virus without symptoms, just with regular T cell response, with interferons and likewise, without ramping up antibodies. But it's not completely free of additional antibodies. There are some antibodies which are there, but at a very low level. So when you get second infection, when you get third infection, that is like repeated vaccination. So immune response will be stronger and stronger with every next time. And every time when we have immune response, we have associated immune reaction. So bodies of the children are becoming capable to react to virus more with repeated infections. So maybe this child was infected first time in the first wave and you never knew. You never tested, and he was fine. But now, 6 months later, he's getting infection again, but because his body is recognizing virus more, it also giving him more symptoms. It is the interplay between human body and the virus. Say, first time when child was infected in April. In this case, we didn't detect any symptoms, but the virus stayed in the body for 2 weeks. Good for virus, but also, okay for child, he didn't care, but maybe was able to give virus to relatives, etc. for 2 weeks, very long period. Second time, when the body already started to clear out virus, we have more symptoms. So say we have like fever, 38 Celsius degrees for 2 days. Now it is detected, the test is positive, but the time when virus stayed in the body shortened from 2 weeks to say 5 days. Not good for virus. Actually, we are fighting virus, not able to multiply for so long. Maybe this child give disease to lesser amount of people around him. So it's all comparable, and also is true the same for older people. There are some people with remarkable immunity, which is very strong T-cell immunity, able to clear out the virus. You probably heard about cases when there is a family, they all live together. When somebody's sick, they care after each other, and 3 of the people get virus, 1 is not. So we in details discussed reinfection. In case of reinfection, we have first infection, then for sure absolutely no virus. And then we've got second infection, and then the symptoms of the second infection could be, depending on the state of immune system, could be stronger, could be the same, could be milder. We observed each of this situation. when it was confirmed by scientific evidence. But there is also another situation which was recently observed. And I myself was actively watching for evidence of this situation for already quite some time. Because, you know, there are viruses like, for example, herpes virus, which is able to sit in our bodies for a very long time, like for years and years. And then when our immune system get weak, for example, when we're getting older, this virus can reactivate and cause a terrible disease called shingles. Yes.
Mm-hmm.
Anybody who heard about shingles or knew first instance what shingle is knows what I'm talking about. So there was always a danger that maybe coronavirus is able to do something like herpes virus does, hide in our body for a long time. Now, from the previous research on coronaviruses, we know that it's very unlikely because other coronaviruses don't do that.
Mm-hmm.
And by comparison, this coronavirus should not do that as well.
For today, I think you've mentioned quite a lot of good points, and people would be clearer now what is the second wave, why we get second infections, and I'm sure people will stay safe and keep protecting themselves and their families. Thank you very much, Antje, for your talk. Hope to see you.
Thank you, Valentina.
Thank you. Bye.
Bye.